Programmed metabolism and diet, which is one of the hallmark of the cancer, reprogramming of energy metabolism. So this glucose metabolic pathway, which is really just breaking down glucose to simpler molecules and to produce ATP. So we have glycolysis, where they break down glucose into pyruvate, and pyruvate can either become lactate or go through more complicated process like Krebs cycle to make ATP. And this just glycolysis, the glucose becoming pyruvate, does not require oxygen. However, the Krebs cycles and the oxidative phosphorylation and then making ATP eventually requires oxygen. But it allows us to make a lot more ATP. So once we go to like pyruvate to acetocoacetate, and then like you Krebs cycle, you get NADH plus and FADH2, and which is necessary for electron transport in the mitochondria, and they produce ATP in mitochondria as well. And the hexokinase is required in making glucose into glucose 6P. So with the initial investment of ATP, glucose becomes GLC6P, which just means the sixth carbon has an ATP, not that it has 6 ATP. And now it became this GLC6P can become both a pentose phosphate pathway or glycolysis, which is a breakdown of glucose to produce ATP, glycogenesis, which is just making glycogen, which is a storage unit of glucose, and others. And there's a thing called Warburg effect, and is the cancer cell tends to exhibit a lot more like glucose than the regular one. Regular cells up to 40 times. So cancer is soaking up a lot of glucose and making a bunch of lactate out of it. And it kind of makes sense that because lactate does not require oxygen, so and cancer is in a hypoxic state, it makes sense for them to produce a lot of lactate. But in fact, they actually cancer cells have sufficient amount of oxygen to go through regular other pathway or Krebs cycles and others. But cancer cell wants to maximize the energy, so they still use up all the oxygen to generate ATP in other ways, and then they use the remaining glucose to produce ATP with the lactate. But that's not really the whole story. The reason why with the hexokinase, glucose becomes glucose 6P, and then glucose 6P is you can make nucleotide, can be used for nucleotide biosynthesis. So nucleotide, there's like DNA and RNA building material, so they can use it for replication of themselves, and the glycerol aldehyde 3P can be used for phospholipid synthesis, so if it's a membrane. So if they're proliferating, they need more membrane material as well. And 3P glycerate, serine and glycine biosynthesis, those are amino acid building blocks of cells. So you go through this glycolysis pathway from glucose to like pyruvate plus NADH, and then we need NAD plus in this process of glycolysis. So the lactate dehydrogenase converts a pyruvate into lactate with a byproduct of NAD plus, and which can go back to the regular cycle of glycolysis. So although the L-lactate dehydrogenase LDH is implicated its function is pyruvate converting into lactate, but the primary purpose or function is acquiring more NAD to keep the cycle of glycolysis going. And once you have this lactate, it can be excreted out of the cell with help of H+, and the H+, because of the presence of H+, it makes its microenvironment acidic. So, and those can be used, the lactate can be used for fuel for other cells, or it could be used for biosynthetic substrate or signaling. Yeah. NAD plus goes back to the glycolysis cycle. So this Warburg effect generates a lot of a lactate. So as this says, it could be a signal because lactate is generated with due to the lack of oxygen, right? Because it's not the most efficient way to generate ATP, but it's just you can do it without oxygen, so that's why you do it. So lactate means a hypoxic signal, which means that it requires more angiogenesis, more blood vessels to transport oxygen, and that causes more VEGF, which is a signal for angiogenesis. So lactate is also subject for energy production, and MCT channel, the channel that how they throw the lactate away is they make the acidic environment. Also, it activates the ECM chain proteases. So in preparation for the new vessel, like angiogenesis, they're weakening the ECM so new blood vessel can kind of like come in there more easily. Extra lactate and H+ also interfere with the immune system. So T cells are glycolytic, which means so T cell doesn't have much mitochondria, so they rely on lactate to get the energy. So, but if they are also using the same process, then first of all, there's like not much glucose in the area, so they can't really use it. Even if you acquire glucose, even if T cell acquires glucose, they can't really throw out lactate and H+ away because on the outer side is already like there's acidic. There's a lot of H+ and H+, so it's harder for them to like throw it out because it happens through diffusion. This is hypoxic, normoxic conditions of when in those conditions what like HIF do, but HIF also serves as a glucose transporter. So HIF combined with MYC serve as glucose transporter, and then they also like expresses or transcribe hexokinase, which helps the glucose glycolysis, and then they also help to express LDH, which we know that it is they convert pyruvate to lactate, shown over here, and also it expresses PTK and helps to keep the scale for lactate generation rather than acetyl-CoA as they are suppressing this. So they all kind of work together to produce more lactate. And there's a thing called IDH, isocitrate dehydrogenase, and when there's a like mutant one, so it's only present in cancer cells, it helps to produce like 2-HG, which is called like oncometabolites, and they are only produced in cancer cells. And it represses prolyl hydroxidase, which stabilizes HIF alpha, which you know is like it's like make VEGF, cause angiogenesis, and all their helps to make more lactate. So it's increase the Warburg effect and angiogenesis. And epigenetics also is affected by like the byproducts of other processes that we've talked about. So AMP, which is a signal for lack of ATP, make AMPK, which is a tumor suppressor. Yeah, something happened there. The NAD+ caused HDAC, but it could be good or bad. But the thing is that like epigenetic changes occur with the various like signals of their others environment. Food, carb, fat, protein, and so it could be for energy, and then it could be used for gene expression. So enzyme like vitamin, mineral, vital for enzyme functions. And antioxidants also are good thing for like cancer prevention and stuff. So carcinogenic contaminants in the food is like PCB in salmon, which is some chemical that's like in farm-raised salmon sometimes. That's known to cause DNA damage. Excuse me. There's preservative or meat cooked in like high temperatures and open flames. They have this thing called N-acetyltransferase, and it's like carcinogen apparently. Dietary deficiency, folic acid, which is like a lot of popular supplement for like pregnant women, but apparently this deficiency is related to colorectal cancer. And then alcohols, there's like 3.5% cancer could be related to alcohol consumption, but for smoking it's like 60%. So smoking, there's probably no such thing as safe smoking, but alcohol maybe there is. Obesity, 15 to 20% related to of cancer related to obesity, which is because increased carcinogen storage, because fat is, they tend to store more carcinogen. Alter hormone metabolism, because like fat people have more insulin, and insulin's kind of like helps to like cells to proliferate, and bad for cancer, or good for cancer, not good for us. Chronic inflammation, cancers love that. Increasing insulin, said that, and also affect gut microbiome, which we'll talk about pretty soon. To talk about the folic acid a little bit more, so the folate deficiency causes either mutation like through this like mechanism, not really sure, but, and then there's also like causes DNA methylation. So, but like without folate, you can't really do that well. So it's hypomethylation, which means like the genes are more tightly packed, and it means it's like less tightly packed. So genes are getting overexpressed, and it could have like oncogene there and then cause cancer. And talk about the obesity again. So there's this like baby mice that was treated with like carcinogen, and then they were fed like normal versus like high fat diet. And normal diet with liver was fine, but the high fat diet showed like cancers. And they looked at the gut microbiome of it, and then a normal diet is like just kind of like looked like this, but the high fat diet tends to have like a lot of green and blue, which is a gram positive. And then so they give like put a thing called BCM, antimicrobiotic, for the gram positive killer, and now there's like a lot more gram negative. And sure, but what they didn't show is not like HFD, high fat diet plus VCM, to see if they cause the cancer or not, but whatever. Cancer preventative factors of diet and exercises. So Mediterranean diet is good, fruit and veggie good, because they're high in antioxidant, as ROS is known to cause mutation, and a lot of vitamin C and E, which known to remove a lot of ROS or antioxidant. They are antioxidant. So vitamin C like use ROS to turn themselves into ascorbyl radical, which is more stable and less harmful than ROS. Similar mechanism of vitamin E. But somehow when you take a bunch of vitamin C and E supplements, it actually like increases cancers a little bit in humans. So we don't know why, but that's the case. The supplement also apparently doesn't really work that well. Next one, the NRF2 ARE signaling pathway. So NRF2 is a master transcription factor. They regulate antioxidant response in cells. And the ARE, antioxidant response element, the promoter region, and for the antioxidant and detoxification enzyme. So when cells are not stressed, you know, there's no reason for them to really make detox enzyme, right? So the keep one becomes doesn't really do much. They get like degraded, and it comes in, and then the NRF2 is keep one like put ubiquitin in the NRF2, and then NRF2 get like degraded. So there's this NRF2 transcription factor doesn't really produce any detoxifying enzyme, whereas cells are under stress, or there's like radioactive oxygen species, then the keep does not cause the ubiquitination, hence no degradation on the NRF2 like here. So NRF2 can combine with MAF to produce detoxifying enzyme. However, at the absence of ROS, but if you like eat like green tea and broccoli, that all somehow like helps to have similar effect to produce detoxifying enzyme, which probably like helps us to prevent cancers. And vitamin D deficiency correlated with different type of cancers, but supplement doesn't really help apparently. There's the really complex figures of the vitamin D3 metabolism synthesis, but synthesized either from like diet or like made because of the sun on our skin. And then like active D, vitamin D is like 1.25 OH2D3. I don't know what that is, but it's an active form of vitamin D, and then it can be used for various things: liver, kidney, intestine, bone, etc. But what they do is the vitamin D help with the regulation of blood calcium phosphorus and Just helps to maintain a good balance. So approved drug for reprogrammed metabolism. So asparagine is soaked up by the cancer cell to make this aspartic acid, and the aspartic acid is like an amino acid, so it's like you can build, it's like just more building materials for cancer cells, so you can like put drug that kind of like interfere with this process, or you can put, so this IDH2M is a mutant, and then you kind of inhibit it, put like drug that kind of like interfere with the process of IDH2.